Hi everyone, this is Jedaidah Chilufya, your instructor, and welcome to the last part of the pre-class week 2. So time flies, and officially we're going to start our class next week. So in this video we'll just talk about the second lab kit that you should start receiving this weekend. And yeah, so I have mine here. I'm just going to go through the things that are actually in the kit. Okay, so one of the things that you find is you're going to get a note which kind of like describes gives like a summary of the things that are in the kit. And so in this second lab kit you have all the items from experiment 2 through to 4. So it's going to give you an idea if you like need it for reference. And then what you're going to get is a digital microscope, and so it's all like still in its like package, and we won't need this until the end of the week. So we'll talk about how to assemble it and how to use it later in the week. So you have that, you can place it separately, but there's actually one item in your kit that needs to be kept cool. So it's in the aluminum foil, and it's actually media. Okay, and so first thing is you want to keep it cool. So it's actually been placed in ice packs, and those should keep your media cool for a couple of days while it's in shipping. But when this gets to your home, please get this media. Okay, remove the ice packs and then get this media and place it in your home refrigerator. So it's gonna have the label of what the media is, the date, and your initials. And so, and how you place it in your refrigerator is that you kind of have to put like upside down. So this is like the right way, right, where the media is at the bottom, but we actually want to place it upside down. So the media, so you have 5 in here, you have 5 different plates. Okay, it's like 1, 2, 3, 4, 5, and they have like red media at the bottom. We'll talk about media during this course. So you want that media to be like on top. That way it kind of like helps to to keep like a steam or like mist from forming. So put it upside down and place it in the aluminum foil. You have to, but just like one is sufficient. And then put this in your home refrigerator. Okay, and then there'll be a time where we need to use it, and that's when you should take it out. But for now, you keep it here. This is sealed so that we don't get any other microbes into the plates. As we know, microorganisms are everywhere, including like in the air around us, and so we don't want any bacteria to kind of like go on our plate. We only want to place the bacteria that we want on this plate. So place that, and in case you're still like traveling or something and you need the ice packs, what you can do is like once you get these, put them in the freezer so they can actually like freeze, get really cold, and then you can place them back the way I did, where like you put them around the media and then you put it around them. You can like travel and keep that like outside, but otherwise try and keep this inside a home refrigerator. Okay, so that's what you have in here. If you have a Ziploc bag which has like what's like extra items, I'll talk about those in a second. Envelope that says plastic pesos, and so if you open that, these will be used later when you start working with the media. So it's just like they're individually packed because they're sterile. Okay, so just keep those in there, and then you actually have some extra ones in this Ziploc bag. Then you have microscope slides. We'll talk about these when we talk about the microscope, but this is what we are going to place your samples to observe under the microscope. So you have microscope slides and glass covers or cover slides that work together to put your sample and then observe under the microscope. Then you also have little tubes that are called microcentrifuge tubes. These we have some extras in here. So these are also going to be used, and they should be like closed. It's like little tubes like that. They're going to be used when you start working with the media. And in there you also have like little labels, like little dots. So these dots, we'll talk about them later, but it's just like, it's because you know how we have to label everything in science. So can actually put our label on like the top of the microcentrifuge tube and also on the side. So these are like little sticky labels that can be placed on top, and then you put your label there. And this actually works really well if you have like a thin pointed Sharpie. If you don't have a thin pointed Sharpie, that's fine. You don't have to use these. You can just like directly label on the tube itself. Okay. So that's that. And then then you have like extra microscope slides and cover slips. Okay, so what's in the extra here? One of the things that we have here is a dropper. So this is kind of like going to help us when you want to like pick up liquid. And so later on when you're working with the media, you actually need to pick up some liquid. And so you also have like a tube that was put in aluminum foil, because it has sterile water. Okay, so we don't want to use tap water because the tap water might have like unwanted substances, but we have a way of like sterilizing water in the lab, and so you have some of that. You're going to need that. And then one thing that I couldn't ship was, at some point we also need household bleach. So any bleach that you have in your home, we have to get that. We have to dilute it and make like 10% bleach, and we use some of this water. So if you want to get like an amount of like a volume, you can actually use these droppers. They actually have like some measurements on here. So you kind of collect that and then put in a tube, mix it with bleach, and we need that bleach for one of our experiments. So we have extra microcentrifuge tubes in here, and extra label that's needed. And we also have this paraffin paper that we need for the media to like seal our media. We'll talk about that when time comes. All right, so that's what we actually have in here. So for all these items, you can just keep them. You can keep them in your box. You can keep them anywhere on your lab bench. They just need to be at room temperature. Nothing special about that. So for me, I'll actually put them back in my box. Sometimes I know it might be possible that maybe the ice packs might have made your box a little wet. So if that's the case, maybe try and dry out the box and then try and put back these items again. And another thing that you have in your kit is a scalpel. It's like we're gonna need this to like cut off some the special structures on the roots, the root nodules, and examine them under the microscope. So we're gonna work on that. And then to observe, clearly observe certain structures under the microscope, you need some kind of like stain. So you have a bottle of this stain. We just need a little bit of this, and this item, because it's a special like lab item. It's something where like after our course is over, it has to be disposed of like in a special way. So we'd have to find a way of like getting it back to the lab for safe disposal. That's what we have for our kit. Yeah, so these items, we'll start using some of them at the end of our first week of class, and then we use the rest of them for the rest of the course. All right. So let me know if you have any questions, or if any of these items are missing from your kit. Let me know. But just remember, you have to keep this cool at all times. So yeah. So for me, I'll actually put this in our lab refrigerator. Enjoy the rest of the weekend, and we'll see you next week for our first week of class.